Yeah, thanks everyone for coming in for the introduction. So the title of my presentation is Nutritional Regulation of Amyloid Beta Associated Single Cell Transcriptomes in an Alzheimer's Disease Model. So Alzheimer's disease is a progressive neurodegenerative disease leading to memory loss and language problems and eventually disrupting execution of basic bodily functions. So the onset of AD is thought to occur 20 years or more before symptom detection. So early diagnosis and prevention is a very important issues for AD. It's a complex disease, so it involves, it's affected by many different genetic and environmental factors. And there's currently no treatment, no effective treatment or cure for AD. So here are the major hallmarks of AD. So the first one is accumulation of these extracellular plaques. So these plaques are made up of amyloid proteins, and they come from this amyloid precursor protein that sits in the membrane, and it gets cleaved by beta and gamma secretases to form these toxic amyloid beta fragments. And so these can aggregate and form these plaques, which can accumulate around neurons. And then the second hallmark are aggregations of these tau proteins that form neurofibrillary tangles inside the neurons. So these tau proteins, they're normally stabilizing these microtubules. And so they become hyperphosphorylated, they dissociate and form these aggregates inside of neurons. And so this leads to a loss of synaptic communication and eventually death of neurons. So there is a lot of risk factors for late onset AD. So the older you are, the more likely you'll have Alzheimer's disease. There's also a lot of genetic variants that predispose you to developing Alzheimer's disease, and the strongest one is ApoE. So if you have 2 copies of the E4 form, then you get a 8 to 12-fold risk in developing AD. And other factors include physical activity, diet, metabolic syndrome, education, and social and cognitive engagement. So AD is often comorbid with obesity, hypertension, high triglycerides, and blood glucose. So these features make up metabolic syndrome, and this can lead to heart disease or diabetes. So because we often see these 2 conditions comorbid, we think that they may be linked. So some of the hypotheses that we have are that AD and metabolic syndrome share underlying mechanisms. AD progression and phenotype is worsened by metabolic syndrome, and metabolic modulation by nutritional intervention, such as administration of metabolic boosting chemicals, could reverse or alleviate exacerbations of AD by metabolic syndrome. So in this study, we're going to be determining the effect of metabolic modulation on 5 times FAD single cell transcriptomes. So our hypotheses are that fructose-induced metabolic syndrome worsens AD progression, and NR and DHA help to prevent metabolic deficits by fructose and enhances metabolic function, leading to amelioration of AD phenotypes. So the mouse model that we're looking at is the 5 times FAD mice. So they're expressing human transgenes that in total have 5 mutations in the APP and PS1 genes. So APP is amyloid precursor protein. There's 3 mutations in this gene, and then there's 2 mutations in the PS1 gene, which encodes for a subunit of gamma secretase, which cleaves APP to form amyloid beta proteins. And so expression of these transgenes and these mutations causes accumulation of these amyloid beta plaques, so that at 1.5 months, you have intraneuronal soluble amyloid beta 42 proteins accumulating inside the neurons. At 2 months, you'll see that extracellular amyloid deposition begins. At 6 months is where neuronal loss starts to begin. And then at 9 months, there's a pretty significant neuronal loss. And so we're taking the single cell RNA-seq at 5 months, so right before there's a loss of neurons. So just going into a brief overview of DHA. So DHA, it resides in brain cell membranes, and it's made bioactive to participate in several processes such as synaptogenesis, neuron formation, cell survival, and metabolic processes such as PPAR signaling. And so their relevance to AD is thought to be from reduction of inflammation, improving blood flow, reducing plaque aggregation, and also maintenance of normal glucose metabolism. So the other supplement that we're looking at is nicotinamide riboside. This is a NAD+ precursor. It's a vital coenzyme for energy metabolism and also for various enzymes, including the sirtuins and the PRRPs. And so it can impact DNA repair, epigenetic modification, inflammation, circadian rhythm, stress resistance, and other processes. And so as we age, levels of NAD+ decline, and this may lead to impairment of many different processes, including proteostasis. And so this could lead to accumulation of these pathogenic protein aggregates. So in this study, we have 7 total groups. We have the wild type, we have the 5 times FAD genotype, this mouse model of AD, and then we have 5 times FAD with fructose, and then we have DHA and NR on fructose plus 5 times FAD background, and also just the 5 times FAD background. And so since I'm going to be mentioning these comparisons a lot, I just spelled them out here. So how to read this is that if we're comparing AD versus wild type, or 5 times FAD versus wild type, we're seeing the effect of AD. And then if we're comparing 5 times FAD with fructose versus 5 times FAD, we're seeing the effect of fructose on AD. If we're comparing 5 times FAD with DHA versus 5 times FAD, we're seeing the effect of DHA on AD, and then NR on AD. And then comparing against the background of ADF, so 5 times FAD with fructose, we're looking at the effect of DHA on ADF and then NR on ADF for these last 2 groups. So the 2 tissues that we'll be extracting are the hippocampus, which is one of the first sites of amyloid beta accumulation. It's involved in memory and spatial navigation. And then the second region that we're looking at is the hypothalamus, which is involved in metabolic control. So this is important for our study because we're studying the contributions of metabolism to these amyloid beta, amyloid beta pathology. And so some of the experiments that we ran, as I said before, single-cell RNA-seq, the Barnes maze test to test for the memory performance, glucose tolerance, and amyloid beta immunostating. So we can see through the immunostating results that there's a maintenance of these amyloid beta plaques in the 5 times FAD with fructose group. So that's colored in green here. So we would expect to see higher amyloid beta staining in the 5 times FAD group. And then with fructose, we're seeing that there's a maintenance of that amyloid beta staining for the CA1 region of the hippocampus and also the cortex. And then it seems with the DHA and the NR that there's a decrease in staining of these amyloid beta proteins. For the Barnes maze test, we saw that the 5 times FAD group and the 5 times FAD with fructose had deficits in memory with the measurement of greater latency to find the escape chamber in this specific type of test for memory. And then we can see that there's a trend towards improvement of memory performance in the DHA and NR groups, but this difference was not significant. So we covered all of the major brain cell types from the hippocampus and the hypothalamus, and some of the expected region-specific cell types, such as tanocytes and choroid plexus cells. So one cell type that I was not expecting to recover that was region-specific was this pro-inflammatory macrophage cluster. So this was distinguished from the major macrophage cluster by S100A genes, CD74, and histocompatibility genes. So we also saw through a differential proportion analysis that there was pretty robust microgliosis of these microglia cells. And so you can see the wild type, that proportion amount is down here, and then these are the AD genotype groups. So there's a lot more microglia being proliferating or that are percolating in the AD groups. We also saw that there was a decrease in the proportion of endothelium cells. So we recovered less of those types of cells, and this might be because of blood-brain barrier dysfunction, which has been seen in AD before. So here I'm just showing the overall DEG results across all the different cell types, the different tissues, and the comparisons. So this is a lot to look at, but this is just to show the overall regulation that we saw with this DEG analysis. And so the major columns are the cell types, and then within the cell type are the different tissues, and then within that are the different comparisons. So we can see right away that there are a lot of DEGs that are being shown in the microglia on the left side here. And then we see other cell type specific DEGs for example, oligodendrocytes, for astrocytes, and for endothelium and OPC cells. And we can also see some cell type shared genes or DEGs that are shared between the different cell types. And I listed some of those here. And then we can also see that the hippocampus and hypothalamus also has similar regulation of genes. So you can see like patterns being repeated between these two tissues. So you can see that for other cell types as well. For example, in the OPCs, we see this repetition of this DEG pattern, and also here in the oligodendrocytes. And then it seems like there are more differences between the hippocampus and hypothalamus for astrocytes and for neurons. And it would make sense for neurons because there's different subtypes of neurons in the two different tissues. So in the leftmost column of these comparisons is the AD effect, and then the subsequent columns to the right are all of the treatment effects. And so we sort of see this inversion effect by all of the treatments. And so I quantified that in this graph, or in this table here. And so here I'm showing for each of the comparisons the total number of shared DEGs. So that's this top row here, the union of the AD signature with the fructose on AD signature, for example. So that total number of shared genes is on the top row. And then the second row are of those shared genes, which are the genes are in different directions. And so you can see that the pattern is mostly reversal for these treatments, and the DHA on AD and NR on AD effects show more of a reversal effect. And then on ADF background, there seems to be also an enhancement of some AD signatures, but for DHA, it's still mostly reversal. So for the fructose on AD effect, there are 3 genes that were in the same direction of AD, so it was further exacerbating the AD effect. And those genes were IL-1 beta, GPR84, and MAN2B1. So IL-1 beta is a well-known pro-inflammatory cytokine. GPR84 is a microglia activation gene, and MAN2B1 has been shown to be upregulated in peripheral cells of AD patients. And then also in the ependymal cells, I saw the upregulation of this complement C3 gene. And it was seen in other studies that this is required for neurodegeneration in AD mouse models. So we can, this may lead to fructose exacerbating a disease-promoting condition. I also saw a prominent regulation of the amyloid beta binding pathway for one of the fructose on AD effects. So there's pretty prominent regulation of this pathway. And then this is for neurons, by the way. And then these are the corresponding genes within this pathway, and they're involved in negatively regulating pathogenic processing and trafficking of amyloid precursor protein. And so this may be a response to increased neuronal accumulation of amyloid beta plaques and seeds. There's also an increase in glutamate receptor expression, which may lead to a more excitatory transmission for these neurons. So here's a figure I pulled from this review paper about neuronal hyperexcitability in Alzheimer's disease. So in the result before that I showed before, there was a regulation of those glutamate receptors. So glutamate is an excitatory neurotransmitter. So it might be contributing to hyperexcitability of those neurons, and that has been associated with AD. And so in this figure, I tried to pull out some of their ideas on what could be happening in this specific feature of AD, and I tried to relate it to results that we've seen. So for example, they pointed out in this figure that there might be hyperexcitability in AD because of the increase of these pro-inflammatory cytokines from reactive microglia. And so we did see that there was an increase in IL-1 beta in the F1AD effect in microglia. And this was down in DHA and NR on the AD and ADF backgrounds. And then here is showing the glutamate receptor. And so this was upregulated in fructose on AD in neurons. And then also in this figure, it's showing in this AD condition, there's higher uptake of these amyloid beta oligomers. And so that might be why we're seeing more of these APP processing and amyloid beta binding related genes being upregulated in fructose, because it might be promoting a state where there's higher uptake of amyloid beta oligomers. So in conclusion, these results may point to fructose promoting a state of neuroinflammation, neuronal hyperexcitability, and increased spread of amyloid beta. And these effects seem to be reversed by DHA and NR. So I'm going to be focusing on microglia, the CNS resident innate immune cells in the brain. So in AD, microglia surround these amyloid beta plaques, and they also phagocytose amyloid beta, so they are thought to have clearance functions of these pathological proteins. And so whether microglia are beneficial or harmful is controversial, because even though they can phagocytose and clear these amyloid beta proteins, they can also lead to their spread. So here are some genes I pulled out. These were in microglia. This is just to show the differential effects of these nutritions. So the first two genes here, they're downregulated, or they're reverse the upregulation by AD in all of these different treatments. And these two genes were associated with late stage microglia or activated microglia. And then these genes here were upregulated, so they enhanced the AD signature, the DHA and NR on ADF background more prominently, enhance these AD signatures. And then for DHA, there was a consistent increase in these TNF, tumor necrosis factor genes, on both AD and ADF backgrounds. And then for these last two genes, it was not affected by fructose, but it was reversed by both DHA and NR on both of the backgrounds. So yeah, there's a lot of specific effects of these different nutritions, and it's hard to make sort of blanket statements on whether it completely reversed or completely exacerbated the AD signatures. So next I wanted to see which processes in a disease regulatory network fructose, DHA, and NR are acting on. So I built a microglia gene regulatory network from using our single cell data, using SING, which is a method that was developed by a former PhD student in our lab. And then I overlaid the microglia DGs onto the gene regulatory network to identify key driver genes whose network neighbors are enriched for the AD DG associated genes. Next, I subsetted the network of using the disease KDs, so took the subnetworks of those key drivers to get a disease subnetwork. And then I ran community detection to get the disease submodules and see if there was over-enrichment of genes affected by fructose, DHA, and NR in those communities. So here's the disease subnetwork that I got, and I annotated the major modules. So in the middle are the microglia activation enriched module. So these genes were involved in myeloid leukocyte activation, lysosome cholesterol homeostasis, platelet degranulation, and chemokine signaling. The blue module also was enriched for microglia activation genes, and its genes were involved in TNF-alpha signaling, neutrophil homeostasis, coagulation, and fibrinolysis. The pink module was enriched for ROVAC GTPA signaling genes, and they were also the homeostatic microglia genes. The brown module was involved in hemostasis, lymphocyte and leukocyte differentiation, FC and B cell receptor signaling, and spliceosome. And then the green module was involved in apoptosis, TNF-alpha signaling, cytokine response, cell cycle, and immune system development. And this bottom module here was oxidative phosphorylation and RNA metabolism. And this was mostly affected by AD, and then the treatments didn't really affect this specific module. So here I've separated out the effects of DHA and NR. So DHA and NR both targeted this central microglia activation module. And then there were some areas where they were being targeted specifically by DHA or NR. So for example, there's an overrepresentation of DHA in the interconnected region between the green and the red modules. And then there was an overrepresentation of NR on ADF DGs in the green module overall. And then also for NR on ADF, there is an overrepresentation of the glycolysis and NAD metabolism portion of the red module, which was also associated with the oxidative phosphorylation module. On the left side here of the network, there's also a enrichment of DHA-associated genes, which was also associated with this microglia activation blue module, and it was regulated by ITGAS and DAB2. So for example, while ITGAS was not significantly shown to be affected by DHA, we see that its downstream effect or downstream genes downstream of these key driver genes were affected. So we can sort of make more inferences on what led to changes that we see in the DHG analysis to further expand beyond just the DHGs that we see through this network approach. So here I'm zooming in onto the red microglia activation module. So here you can see that there are context-specific effects of DHA and NR. So for example, you see only like the blue being colored here for some of the nodes, and only the green being colored here. So both the blue and the green colored nodes are the effects of NR, but they're on different backgrounds. And so we can see that in the different backgrounds, we see different effects of these treatments. So for example, the effect of NR on AD shows a downregulation of these microglia activation genes. So these are genes that are upregulated and activated or reactive microglia, whereas there was a specific regulation of these glycolysis NAD metabolism genes only on the NR on ADF background. So this might be because in a state where we're trying to metabolically challenge the mice with fructose in this already sort of vulnerable state of AD, NR might be recruited to serve additional functions to play in increasing like alternative sources of ATP generation, which would be glycolysis. And then on this side, there was an increase of these homeostatic genes by DHA and NR on the AD background, but not on the ADF background. And also looking at the regulation of this module, for example, this gene here, CST7, was affected by a lot of the different effects. And it was predicted to be downstream of a lot of other genes. And so this might be because of effects in other parts, other genes in this network, and through network propagation, it all sort of converged onto this specific gene here, and that might be why we're seeing more prominent changes in these genes because it's affected by a lot of other genes. And so, yeah, a lot more to dig into, but basically our network approach, we're able to infer more of the mechanisms that might be happening for us to see these differentially expressed genes. So we didn't have a lot of fructose affected genes to begin with, but one small area that fructose affected was this here between the red and the green modules. And this included the GPR84, IL-1 beta, and MAN2B1 genes. And interestingly, these were the same genes that were showing an exacerbation of the AD effect, where there was an upregulation of AD, and it was further upregulated in fructose on AD. So it's interesting to see that they're interconnected, and GPR84 acts upstream of IL-1 beta and MAN2B1. So next we wanted to do subclustering of the microglia. So we identify 5 reactive clusters and 2 homeostatic clusters. And here I tried to list some of the distinguishing markers between the reactive subtypes. In all the treatments, there was a loss of this cluster 4, which was associated with AD. And then in the DHA and NR on ADF background, there seemed to be more of this cluster 3 subtype. So showing sort of an enhancement of this AD associated cluster. So that might be where we're getting the enhancement of certain AD signatures by DHA and NR on the ADF background. So next I ran pseudotime analysis to see the transitions between those different subtypes. And so that cluster 4 was predicted as a terminal cluster, 3 as an intermediate, and 1 as sort of a bridge between the homeostatic and the reactive clusters. And the genes that were changing along this trajectory, I listed here on the, or plotted here on the right side. And so I broke the genes down into 4 different groups. So the first group are genes that are activated across all reactive microglia. And then group 2 genes seem to be ones that seem to turn on towards the later stage of this trajectory. And then group 3 are homeostatic microglia genes that are turned off by the activated microglia. And then group 4 genes seem to be genes that are expressed across most microglia, but seem to be turned off at the later stage of this trajectory. So we retrieved a lot of different cell type specific transcriptomic signatures, and we wanted to see if they had any relevance for human AD GWAS. And so we ran mergeomics to see if any cell type function, so that would be the marker genes, any cell type specific processes, so those network modules that I got from the SING gene regulatory network, and also if any of the DEGs were enriched for AD GWAS. So using the marker set enrichment analysis, I saw that AD GWAS signals are enriched for these immune cell types, so microglia and macrophage marker genes, and also oligodendrocyte marker genes. I also tested for enrichment of the pseudotime groups that I plotted earlier, and there was also very consistent enrichment of these AD GWAS signals across all the 6 GWAS that I looked at for AD. And then here I listed some of the SNP mapped genes that were part of the gene sets and that were pulled from the GWAS. So looking at the results for across the different DEGs, I see that there is cell type and comparison specificity of DEG enrichment of AD GWAS signals. So for example, hippocampal parasite AD DEG show a high enrichment of AD GWAS. The DHA on AD DEGs of endothelial cells show high AD GWAS enrichment across both of the tissues. And then across all of the different cell types, it seems that DHA on ADF comparison DEGs were highly enriched for GWAS. signals for AD. And then the last thing to note is across all the 6 different GWAS studies that I looked at, the neuronal NR on ADF genes were highly enriched for AD GWAS signals, and these included some well-known AD risk genes such as ApoE, CLU, and SORL1. So looking at the CINCH module enrichments across a lot of the different modules I had gotten for these CINCH networks, there's some very consistent enrichment of these AD GWAS genes. And so I listed some of those here. And further exploration of these CINCH modules can provide us some insight into which cell type processes are relevant for increasing disease risk. But yeah, I'm still very much looking into this still. So some conclusions that I had here are that fructose, DHA, and NR are able to modulate genes important to AD and microglia. Fructose reverses some microglia activation genes, but exacerbates certain AD-associated inflammatory genes and may promote a pathological neuronal state. DHA and NR show some context-specific effects. And interpretation of these changes of the AD signature by fructose, DHA, and NR is challenging. There's just a lot of different ways that you could interpret these changes in terms of whether they're harmful or beneficial. Yeah, and that's the end of my presentation.